Hello everyone! We have come to our final chapter for the course Bio 3201, Chapter 11, Application of Genetic Technology. A major milestone in genetic research is the use of recombinant DNA technology. So what is recombinant DNA technology? Recombinant DNA is made by combining different fragments of DNA. Usually the fragments would be from different sources, from different species. So because they are unrelated genes but they are combined together, they are called chimeric DNA. The recombinant DNA technology came into existence in the 1970s. So it has been a whole revolution in biochemistry, in genetics, in all the life sciences. It all started with the discovery of restriction endonucleases or restriction enzymes by Arbor, Smith, and Nathans in 1970. So it's a very important tool that we have in genetic technology. So these restriction endonucleases are found in bacteria. They actually protect the bacteria itself against viral infection. So when a virus infects a bacterial cell, the restriction enzymes actually split the viral DNA. So the viral cannot replicate. Thus, the virus is destroyed. When So we have hundreds of restriction enzymes that have been discovered so far. They are named after the bacterium in which they are found. So we have like E. coli, Hein 3. Those are all based on the bacteria. So recombinant DNA technology has evolved to be very valuable in medical sciences, diagnostic treatment of diseases, uh, what we are currently facing, the coronavirus, is using this technology, genetic technology, to actually detect the presence of viruses, like what you did in your infographic assignment. And then we have gene mapping in agriculture, animal livestock husbandry, various industries. Um, and let's have a further look at some of the major applications of our genetic knowledge and technology. Advances in medicine. Uh, Due to recombinant DNA technology include the mapping of the human genome. So we have methods such as restriction fragment length polymorphism, or RFLP, that uses restriction enzymes like we discussed earlier to actually detect variations in DNA fragment length. So this can be used to actually distinguish between plant species, genotypes, and in some cases, the individual plants themselves. And then we have DNA fingerprinting, also known as DNA profiling. I bet you all are familiar with this. Is a technology that identifies individuals using properties of their DNA. So previously, this method uses variable number of tandem repeats of VNTRs to distinguish between the individuals based on the unique pattern of band on a gel. Remember your electrophoresis gels? So, but right now, currently, uh, new automated DNA fingerprinting technology uses PCR to amplify short tandem repeats, or STRs, or microsatellites to produce patterns of peaks instead of bands in gel. So DNA fingerprinting is important in medicine, uh, like I said, in our current coronavirus situation, as a diagnostic tool to identify the virus, and in forensics to provide evidence at a crime scene, and also in relationship testing, as in identifying a child's biological father or mother. Okay, so next we have gene therapy, which is basically the introduction of clone genes into living cells in an attempt to cure disease. So it represents a new potentially powerful means of treating a wide variety of illnesses. But of course, research is undergoing. A key step in gene therapy is the introduction of a clone gene into the cells of people through either non-viral or viral gene transfer methods. Okay, and something that is more current right now, the latest technology would be CRISPR. Have you ever heard of it before? So CRISPR-Cas9 is a unique technology that enables geneticists and medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. So it's real time. It is currently the simplest, most versatile, and precise method of genetic manipulation, and is therefore creating this big buzz in the science world. But of course, the research is still undergoing. We do not know the effects of this yet. Next up, biotechnology in agriculture. So biotechnology holds this big promise in producing transgenic livestock, transgenic or genetically modified organism, GMOs. And one very interesting method would be molecular farming. So it's a P instead of a F in farming. So it's a novel avenue of research that involves the production of medically important proteins in the mammary glands of livestock. So has various advantages. Uh, First of all, compared to the production of proteins and bacteria, one advantage is that certain proteins are more likely to function properly when expressed in mammals. So this may be due to the post-translational modifications, for example, attachment of carbohydrate groups that occur in eukaryotes but not in bacteria, so cannot translate. Next advantage would be that certain proteins may actually be degraded rapidly or folded improperly. You know your proteins have to be folded, so they may be folded improperly when expressed in bacteria. And then of course in terms of yield, when we have a cow, compare a cow mammary gland and a bacteria, of course, it would have higher yield of the recombinant protein. Let's say we have a transgenic cow. It can produce around 1 gram per liter of transgenic protein in milk. So this is an example of what has been done. So we have a human hormone gene here. So using this recombinant DNA technology, it's cloned. The human hormone gene would be cloned next to a sheep beta-lactoglobulin promoter, and this promoter is functional only in mammary cells so that the protein product is secreted into the milk. So that's what we want, protein inside a milk. So it's transferred into a plasmid vector, and this plasmid vector will be injected into the sheep oocyte so that the plasmid DNA will integrate into the chromosomal DNA of the sheep in addition of the hormone gene into the sheep's genome, and then implant back into the fertilized oocyte into a female sheep, which then gives birth to a transgenic sheep offspring. So from this transgenic sheep offspring, um, you can actually obtain milk from the female transgenic sheep, and it would contain a human hormone. This will be milked and purify the hormone for medical uses. Next up, we have transgenic plants. So plants can be given characteristics that are agriculturally useful. For example, the company Monsanto, very famous for GMO corn, uh, they have produced transgenic plant strains that are tolerant to glyphosate, which is the active agent in the herbicide Roundup. So compared to non-transgenic, the plants can actually grow in the presence of the glyphosate containing herbicide. So the plants grow even though they are sprayed with herbicides. And then disease resistance in plants. We have like virus resistance, which have been developed by introducing a gene that encodes a viral coat protein. So then when the plant cells express the viral coat protein, they become resistant to the infection of the pathogenic virus. And then of course we want to improve shelf life. The company Calgene Incorporated have developed the flavor saver tomato. So this transgenic tomato plant has been given a gene that encodes an antisense RNA for polygalacturonase in mRNA. So the enzyme polygalacturonase involved in the ripening of fruit, it digests sugar linkages within the pectin found in the plant cell walls. So the antisense RNA binds to the polygalacturonase mRNA. It prevents it from being translated and degraded. So the tomato do not get spoiled as quickly as traditional tomatoes. The very basic applications of genetic technology. Genetic research is making new advances every single day, so there's always something new out there. But with this, it's the end of our course for Bio 3201. Before we end, I would just like to share a quote that is relatable to our current situation right now. So don't let what you cannot do interfere with what you can do. That's all from me for Bio 3201. Assalamualaikum warahmatullahi wabarakatuh and good luck for exams. Bye.